Hey y'all, it's Jafar, your TA. I wanted to go over a few concepts more than a few, for the final exam that I feel like y'all might struggle to understand, and I wanted to give like more of a lesson on it because we didn't have much time in a few of our labs for the last few weeks. So yeah, so for the Bradford assay, that was our first lab after the midterm. We looked at two variables, right? So in our chart, when we made that chart, we looked at absorbance on the y-axis, and then on the x-axis, we had concentration, okay? Conc. All right. So the slope for that looks something like this. So when it looks like this, that tells you that you have a directly proportional relationship, okay? So this means that when one thing goes up, the other thing goes up. So they're directly proportional to each other. So if you have a higher concentration of something, then you have a higher absorbance, right? That makes sense. Okay. And this is proven by Beer's law. Okay? So you need to know Beer's law. And it's summed up in that equation where absorbance equals molar absorptivity and then path length and then concentration, okay? All right. The spectrophotometer, I went over the function of that a little bit. Essentially, so you have your sample, right? And then protein, blah, blah, blah. So what the spectrophotometer does is it passes light through the sample, and then it reads whatever light passes through the sample. It reads basically what light didn't pass through the sample. Okay, so yeah, it kind of takes that difference. So that's all you really need to understand, is that the light passes through the sample, and then some device at the end reads how much light is got absorbed by the sample. Basically, how much light didn't make it. So the dye, we used a dye for this lab as well, Komasi blue. Remember I said, so if you have a protein, right, the way this dye kind of has a universal effect on all protein in a sample, right, because all the proteins look different, right, is it binds to, I think, 4 amino acids. You don't necessarily need to know the amino acids themselves, but just know that the dye binds to these amino acids throughout the protein's primary structure, right? See, like this, the dye was like purplish blue. So it binds to different amino acids throughout the protein, and it binds, and every protein should have at least one of those amino acids, right? So, yeah, and the wavelength for this dye is 595, and this wavelength in particular is the wavelength, so lambda, at which you get a peak absorbance. So basically this is the wavelength where it's most sensitive and will give us like the best estimate. So that's the importance of this wavelength. Yeah. So, and last thing with this lab is the purpose of a standard curve. So the reason we do standard curves is to get that. So right, we do a standard curve, and then we get a slope formula, right, where the y and x are your values on each respective axis, right? So using this slope formula, we get a slope value and an intercept. And we can use this to find an unknown. As long as we know either the x or the y value of that unknown. So in our case, we knew the absorbance of our unknowns, but we did not know the concentration. And so that's what we had to calculate. So we want to find an unknown value using our standard curve. When we do our standard curve, we get a slope formula, and then we use that slope formula to calculate either the x or y value for an unknown. Okay? All right. Okay, so I'm moving on to the other labs. So we did protein extraction. Okay, so you remember that short lab where you just put the fish muscle in the Lamley buffer, and then we were just gonna wait for it to break down, and then the next two labs we did the Western blot and gel electrophoresis, and then we did immunodetection. Okay, so for protein extraction, some concepts I want to hone in on. So the proteins need to be denatured before SDS page. So SDS page is the gel electrophoresis process itself, right? SDS is part of that buffer, but SDS page is a process, right? The GE in page is gel electrophoresis. So SDS, sodium dodecosulfide, polyacrylamide gel electrophoresis. That's what SDS page stands for, right? And the reason you want to denature before you do SDS page, right, is if I have a protein and it looks like this, right, I'll fold it up nice and neat in its tertiary structure, right? That might affect how it passes in the gel, right? You might have some of this protein here, some of it here, some of it here, right? It might not process correctly, right? So you want that protein to be in its primary structure, right? So all loosened up and denatured, right? This will give it sort of a uniform effect to where it'll land in only one spot. Okay, so essentially, you want to denature the proteins because a renatured protein might affect the travel length of the gel and make it less consistent. Okay. All right. And I hope you're taking notes while watching this, by the way. Of course, this is a video, so you can always run it back. You can play my voice at 1.5 speed for all my ADHDers watching this. But yeah, so this one's a big one. I want to go over this one because I feel like I kind of was lacking on this explanation, and I definitely want you to know this before the final, okay? So the muscle organization, so you have a muscle, right? And this can vary on the organism in terms of complexity, right? So this is a dissection of a muscle. So muscles, the way the tissue is organized is you have bundles within bundles within bundles, okay? So you have here, I'm not going to go over the exact terms. I don't care that you know them. You can draw on the final as long as you have an explanation to go with that drawing. But essentially, you have these bundles that contain bundles in them, right? So I'm going to zoom in on one of these. So in here, you have muscle fibers, which are also muscle cells. Okay? So within this muscle cell, you have what's called sarcomeres. The sarcomere is the functional unit of a muscle cell. Okay? So I'm gonna zoom in on this sarcomere, right? So the sarcomere looks something like this. Okay? So, within the sarcomere, you have thin filaments, also known as actin, and you have thick filaments, also known as myosin. So the actin is attached to the Z line or Z disc, right? So I'm drawing these red lines, that's your actin. And then your myosin is the thick filament, and it attaches to the M line. And I'm going to draw this in a second. So this purple here is your myosin. So in the middle here, we have the M line. I'm going to use a different thickness. Here's your M line. It's easy to remember because it's in the middle, right? And then your Z disc kind of looks like a Z shape, right? Like that. So that's also easy to remember, I hope. So Z line, Z disc, I've heard a couple different things. As long as you say Z. Yeah. And then here's your actin. So I want you to be able to draw something like this. If you can draw something like this, then you're good to go. Okay. Myosin. And then here's your M line. Okay. So essentially what muscle contraction is the actin filaments getting closer to the M line. That creates this shortening effect, allowing the muscle tissue to shorten and contract. And that the larger effect you see is movement, usually. Yeah. What else do I want to go over? Yeah, this one's a big concept right here, so understand this. All right. The proteome, I should have talked about this. This is an important word. Each of our labs after Bradford assay was called proteomics 1, 2, and 3. So the word proteome refers to the entire set. Guys, sorry, I have cats fighting. The entire set of proteins within, now this can refer to within a cell, an organ, or an organism. A cell, organ, organism, whatever. At particular conditions. So at certain conditions. Okay? If you can write something like this and understand it, then you know what a proteome is. Okay? Yeah. Okay. I want you to also be able to draw a myosin protein. So what a myosin protein looks like generally is like this. So you have a coiled coil structure. I don't know who named that, but I want to smack them. Like something like this. Right? Pretty. Okay. And then this, what I just drew here, is the heavy chain part of the myosin. Okay? So this is the heavy chain. Then on the neck region of the myosin, you have LOL. You have these light chains. This is actually what we looked at in our experiment. These light chains, the red and green that I just drew. So here, this is your light. Oh, sorry. I was just watching Death Note. Light chains. Okay? Alright. And then this region here, where the light chains are attached, this is called the neck region. Right? And these two are the heads. Okay. So let me take a look. All right. Here on the head, on each head, we have important places. So up here we have the actin. I'm gonna use a different color. This is the actin binding site. And then somewhere here, about right here, you would have the ATP binding site. This is where the ATP gets broken down, and then you get that energy that allows myosin to cock the head and then pull the actin back. Okay. This is pretty much all I want you to know. I'm not going to go into it too much further. So on the head, you have the ATP binding site in yellow, actin binding site in orange, the light chains on the neck region, and then you have the heavy chain, which is like the main part of the myosin. Okay? All right. Moving on. Let's look at immunodetection. All right. So I explained this pretty thoroughly in class, but just to make sure that everybody kind of knows or wants to just review, down here you have the protein of interest that you're looking at, right? In our case, it's called myosin, light chains, okay? But you can just say myosin. And then, so here's our protein, right? This is on the Western blot, on that paper, the membrane, the nitrocellulose membrane. So on here we have our protein, right? It's invisible to us right now until we did our treatment. So what happens first is that your primary antibody binds first, okay? And then what binds to that primary antibody is secondary antibodies. Could be 1, 2, I don't know how many is the cap, but like multiple for sure. So you have a secondary antibody. Okay, and I'm just drawing it smaller. I don't know if they're bigger than each other, but yeah. So, and then attached to this secondary antibody, you have an enzyme. I like to draw enzymes like Pac-Man, okay? In our case, this enzyme is horseradish peroxidase, right? Because it breaks down hydrogen peroxide. Peroxidase. Okay. Yeah, HRP. And then the substrate of this is I didn't go over the name for this. I think I briefly mentioned it, but it was 4CN. It's like 4-chloro something. Y'all are taking OChem, so you'd probably be able to draw it, I think. But anyway, so you have 4CN, gets broken down and produces a color. Okay? Or actually, I don't know the exact mechanism. I don't care for you to know the exact mechanism, but the hydrogen peroxide, I'm pretty sure, actually gets broken down, which interacts with the 4CN. So you can, just for the sake of ease, just you could just call this 4CN. And then when I did immunodetection, we would just use an actual substrate that gets broken down, and that makes a color. So it just depends on what you're doing. But yeah, the color we're using here is called 4CN. Okay, so secondary antibody right here. And then this is the primary antibody. Okay? Alright, so what's happening here is that you have a enzyme substrate reaction. Okay? Enzyme substrate reaction. That's what's happening between these two components of immunodetection. Okay? Okay. Next. All right. One more concept I want to go over with this. So the reason we do secondary antibody attached to the enzyme and all instead of just doing the primary attack, like the reason we don't have this. Actually, you know what? I'm gonna draw like a comparison. The reason this is less efficient, right, is because over here with the secondary antibody, you can have a stronger signal because you can have multiple secondary antibodies with that enzyme attached on one primary antibody. This gives you a stronger signal. That's ugly, but you get the point. All right. So you get a stronger signal with this over here rather than this. All right. So stronger signal because, or you could say signal amplification due to a higher number of secondary antibodies that can bind to the primary antibody. So something like that. Okay. But as long as you understand this concept right here, so why this is typically more used in a lab setting. Okay. And if you have like a protein that's not very present in the in your sample, then this will help amplify that signal and really show you that it's definitely there. It's just not that much, you know? All right. So next concept, stop. Next concept I want to go over is the kaleidoscope. Okay, so you remember in that gel you had one well where it was multiple colors? Something like this, right? Very pretty. Right? So the kaleidoscope is made up of covalently dyed whatever. I spelled that wrong. Covalently dyed proteins with known masses. This allows us to say, okay, at this particular distance, we know that any protein with the mass of, let's say, 25 kilodaltons, right? At this distance, your protein should be about 25 kilodaltons, right? This green one, for example, right? So this gives us like a standard set of proteins to compare to and allows us to estimate molecular mass of unknown samples. Whether it's visual estimation or like we did with ImageJ, right, where we digitized it and we took the exact pixels and everything. Okay, so that allowed us to, like, say we had a myosin here of a tilapia, and we could say it's about 25 kilodaltons, right, or a little above, for example. Yeah, so that's the purpose of the kaleidoscope. 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 All right. New page. So a lot of you got confused with this. Willie. So the positive and negative controls. So in our case, the positive control. One sec. Was myosin. And the negative control was actin. Okay, and this is referring to on the actually I drew a really pretty one, so why is this so pretty? Why waste it? On the last well, we had those two controls here. So on this well, right? So what we wanted to see was that a myosin band show up, but not an actin band. Okay? We're just like dash it here. Okay? We don't want to see that actin. Actually, let's draw it more like this. Here's our myosin band. Actin should be lighter, so maybe like right here. So the difference between a positive and a negative control is not. This is not about charge at all. Okay, this is an experimental term. Essentially, a negative control is a group or condition where you expect no effect. So that's actin. So we don't want to see a band. If we see a band, then that means that our antibody is not specific to myosin. That it can be specific to other things like actin, right? So that's bad. So a negative control is a group or condition where you expect no effect. So I'm gonna write that down. Negative control. Expect no effect. And then a positive control is the opposite, where it's a control group where you do expect an effect. This is good to understand for like just general research understanding when you're reading like a paper and they're saying this was our negative control, this was our positive control, right? A positive control is a group or condition where you do expect a known effect. Okay, so that's our myosin. We do expect to see a band show up because we expose this membrane, the nitrocellulose membrane, to antibody specific to myosin. Okay, so there was some confusion on that on the quiz where people thought this was referring to charge, that actin was negatively charged and myosin was positively charged. No, that has nothing to do here. Okay, yeah. So I hope y'all are still paying attention by now because all these concepts will be on the exam. I can promise you that. Yeah. So, y'all, I want y'all to understand why we took the log 10 versus just the mass. So, in immunodetection, we did y coordinate or the distance in pixels, right? And then here, we took the log 10, or we put the log 10 of the mass. In our y-axis, okay? Y, x. So, understand which goes where. So, the slope for this looked like this. And this makes sense, right? Because the heavier the protein, the more weight, the less distance it should cover, right? So here we had larger mass, right? Covers less distance here, okay? And then smaller mass covered more distance, right? That makes sense, okay? So this relationship where the higher one value is, it makes the other go down, is called inversely proportional. Okay? This is an inversely proportional relationship. And the reason we took log 10 mass instead of just raw mass here is because if we did the regular mass, right, in kilodaltons. Shh. If we did the mass, and then here was our y coordinate, then actually the relationship here would look something like this, right? Where it's not really linear. Okay. And so because of this, we wouldn't have been able to get a slope formula, y equals mx plus b. Okay. So, and because of this, we would not be able to estimate unknowns. Okay. At least not numbers wise. We can estimate them visually, but to get exact numbers, we would not have been able to do this if we took the raw mass with the y coordinate. Okay, so we had to convert it to log 10, and then remember we can. Okay, and then we converted it to back to mass with the inverse log. So yeah, so this is why I'm showing you here. This is why we took the log 10 mass and not just the regular mass. Okay. And the relationship between log 10 mass and the y coordinate is inversely proportional. All right, making sure I'm covering everything. Okay, the reason we do a Western blot generally is to detect the presence of a protein. Okay. And the reason we do this detection on a Western blot and not gel electrophoresis is because with gel electrophoresis, I'm abbreviated as GE, we cannot expose it to antibodies because the gel gets in the way, right? Cannot use antibodies, and antibodies are specific to what we're trying to look for, right? So cannot use antibody. Yeah, cannot use antibody on a gel. So we do a Western blot. We blot it on a nitrocellulose membrane, which allows the antibodies, the very specific antibodies, to specifically bind to what we are trying to look at. Okay, so yeah. So these are two reasons I want you to know. So why do we do a Western blot? To detect the presence of a protein. And why not just do it on a gel electrophoresis? It's because we cannot use the antibody on the gel. Okay? Right. I think, yeah, that's all the concepts I wanted to look at. Oh, one more thing. The reason we extract muscle from different species of fish is to check if they're statistically significant from each other. Statistically different from each other. Okay, significantly different from each other. Whatever. We just wanted to see if the weight of the myosin light chains was different between the tilapia and salmon using numbers, right? We looked at it scientifically. So that's the reason we wanted to look at muscle from different species, was to see if there was a difference, a statistically significant difference between the myosin light chains weight of the salmon and tilapia, respectively. Okay? Yeah, without further ado, I think that's all. Have a good Thanksgiving break. I hope y'all look at this video before the final, and this should definitely help you a lot with most of it. So, yeah. All right. Have a great day.